Today is a very interesting day for us, a very important day for us. It's the first day that we are running a real-life sample for a student at the University of Stellenbosch on the Oxford Nanopore MinION instrument. It's additionally interesting because the sample in question is Nguni Sheep, which is an African breed of sheep, and we're trying to create the genome sequence for this sheep.
I am from the Animal Science department and I'm registered at Stellenbosch as a PhD student. What my project is based on is the sequencing of Nguni Sheep of South Africa. So the Nguni Sheep, they are divided into three groups. We've got the Zulu, we've got Pedi, we've got Swazi. So what we want to do is we want to sequence their genome so we can be able to compare their genotype with other breeds from other continents like European breeds and also the Asian breeds.
Diplomix was set up about 5 years ago with support from the Department of Science and Innovation through the SAREA program. And our mandate is to increase productivity in the omics technologies in South Africa, to improve the infrastructure and general system productivity. So a lot of that is training and a lot of that is infrastructure development. So it's really great to be here today as part of the first run at CAF on the Oxford Nanopore. This is a great example of where the technology can be used for research groups in South Africa, and we're really excited about the Nanopore initiative and the projects that we're going to be supporting going forward.
The Nanopore instrumentation is very good at producing long reads of sequence data, and that's very important when you're working with more complex genomes like mammalian genomes and even large fungal genomes. Plant genomes, of course, as well. These long reads allow you to jump over repetitive sequences or sequence across repetitive sequences in genomes, which is very difficult to cross with short read technology. For a long time, one of the big holes in our ability to service our clients was when we tried to look at whole genomes. And this is definitely bridging that. The technology is also very amenable to actually move to sort of as an everyday tool into labs. So in some instances, people might prefer to use this instrument in their own lab instead of using it within a service facility. And that's an important step in the evolution of sequencing technology for us.
The platform is going to assist South African technologists and animal scientists, more to be specific, the molecular biology side of it. That is because, as I've mentioned before, it's the latest technology and it gives the long reads, which is easy to align, and it gives you proper information.
Things like short read technology becomes more difficult to employ, and gaining information out of the data itself becomes more difficult. And these long read technologies make that process a little bit easier to actually gain information out of pure data.